Hello and good morning from New York City. To all of our viewers, thank you for joining us. I am Sarah Demy, and on behalf of the Demy-Colton team and our sponsor for this virtual salon, WBB Securities, it is my pleasure to welcome you to the 39th installment of the Demy-Colton Virtual Salon Series. The title of today's salon is Candida Auris, Ignore It and Other Emerging Pathogens at Your Own Risk. So we welcome everybody back who has joined us in the past, and if you are joining us for the first time, thank you very much. We hope to see you again in the future. As I've said in the past, Demy-Colton is all about convening events that bring people together to advance the biotech industry. And we do that through our in-person events, which include BioFuture, which will take place here in New York City, October 4 and 6. Biotech Showcase, which takes place alongside the JP Morgan Healthcare Conference out in San Francisco, as well as our virtual salons and our CEO summits, an invitation-only event that is taking place in Barcelona in June and in La Jolla in July. As I noted up above, the topic of today's salon is Candida auris, Ignore It and Other Emerging Pathogens at Your Own Risk. And before I get underway, I do, again, want to thank our sponsor, WBB Securities and Research, for sponsoring this event and for bringing together a great panel, or two panels, as we'll see. Before I turn the virtual mic over to the moderator, I want to let you know that the formal discussion will take place in two segments. The first will look at Candida auris and emerging pathogens, and the second segment will focus on the risks and the solutions. The entire discussion will last about 50 to 55 minutes, and it will be followed by a Q&A session. For everybody who is out there, please note that this is going to be recorded and it will be available for on-demand viewing a little bit later on today. And you can access any documents or materials that have been provided by our sponsor or our other panelists on the Event Resources tab. Now to today's salon. I am pleased to introduce our moderator, Steve Brozak. Steve is the managing partner and president of WBB Securities. He will be joined by Pravabati Fernandez, chair of the National Preparedness and Response Science Board, Robin Robinson, former founding director of BARDA, and Tom Walsh, founding director of the Center for Innovative Therapeutics and Diagnostics. So without further ado, it is my pleasure to turn the podium over to Steve, and I will return again to introduce the second segment. Steve, the stage is yours.
Thank you. Thank you to Biotech Showcase and Sarah Demy for organizing this important salon. I'm Steve Brozak. I am the managing partner and president of WBB Securities, but I'm also the founder of WBB Research Institute. This is the anniversary of the launch of WBBRI, which we created to bring attention to critical healthcare concerns. And in keeping with that, today's salon discussion will cover the crisis we face, specifically with antimicrobial resistant pathogens, and of course, Candida auris. Now, we thought about starting this salon with a trailer from the current HBO series, The Last of Us, which deals with a worldwide fungal apocalypse. But I think this is one of those times when reality is frankly more frightening than even a good Hollywood fungal zombie movie. But with all levity aside, I want to make sure we dive directly into the questions and discussions. So I want to introduce the remarkable first panel, which will focus on how did we get here? And with that, let's start with the first participant who will introduce herself, Dr. Prava Fernandez.
Hello, Steve. Thank you for having me on the show. I am the chair of the National Biodefense Science Board and have been in anti-infectives for over 35 years and have been involved with antibiotic discovery and work on antifungals too. So let's dive into the matter as to how we got here. Having been in this space for so long, something like over 40 years in this space, we've seen antibiotics being discovered over a period of time. And now we have a lot of generic drugs, which actually work in many cases. However, new drugs, new antibiotics are needed, both for bacteria and fungi. And as resistance happens, and a few people do get infected, especially those who are aging, who are immunocompromised, who have stem cell transplants, or for some other reason have a compromised immune system. The problem with the new medications are when an infection happens, they get used, but it's a few cases. And the companies which have strived long and spent a lot of money just cannot have returns on their investment. So they're having a hard time in managing those companies. Many of them have failed, and many of them have found strategic partners. Now, we need the new antibiotics. And why is it different now than it was before? It now costs much more to actually have an antibiotic approved by the FDA because of new guidances which were established right at the start of the century. And they take many more patients to enroll, cost a lot more. So it costs more, you make less money, so it's very hard to develop a new anti-infective. Now, in the case of antifungals, for instance, if you need to find a drug for Candida auris, it is even more difficult because you have to enroll these patients, find them, and if they are found, since these fungal infections are really in immunocompromised patients and rather sick patients, if you look at all-cause mortality in these patients, they could be dying of something else other than Candida auris who could have gotten treated. So it's very hard to run a trial for some of these fungal pathogens, especially, to enroll them and then to have a successful trial. And of course, then there are fewer patients, and it's a problem that there are not enough to treat. However, the number of cases are increasing. I'm currently in Geneva, and I just heard at a presentation today in a neonatal study in one of the lower middle income countries, 18% of neonates who died had Candida auris. So it is spreading, and if it happens there, it will happen here.
Prabha, thanks for that sobering introduction. Well, with that, I'd like to next bring Dr. Robin Robinson. Robin has had a distinguished career and is currently the CEO of Asperovax. But Robin, why don't I let you tell the viewers about yourself, and then I've got a quick question for you, please.
Thank you, Steve. Good to see you again, Prabha. I am, as Steve said, the Asperovax CEO, where we work on oral RNA vaccines and therapeutics. And I was at BARDA for a number of years, and for 8 of those, I was the director, and started the whole process there at BARDA through 2016. This actually reminds me of what we saw in 2013, 14, 15, 16, 17, and all the way into 2020, with epidemics starting with a hockey stick type of trajectory. And if you look at the numbers that the CDC has published as they rise in 2021, 22, and in 23, it's straight up. And I remember very much what we were seeing in West Africa with Ebola, and certainly in 2015 with Zika, that its trajectory is very, very similar. And of course, what we've seen more recently with the COVID-19 pandemic. And I'll talk a little bit later about how my former organization, BARDA, came to be involved with antifungal after a long, torturous road that explains a little bit of the government bureaucracy, but also of being able to jump out ahead of this and with the infrastructure that's needed. With that, I'll turn it back over to Steve. Thank you.
Thank you, Robin. Thank you very much. We have Dr. Tom Walsh, who has a direct dial into us. Let me introduce him and Dr. Walsh. Tom, if you could give us your background, and I'd like to, as someone dealing with the front line of infectious disease, I've got a quick question for you as well. Dr. Walsh. Okay, well, looks like Hollywood media is not working here. Tom, can you hear me? Oh, here we go. Tom. Tom, are you there?
Number you have dialed is unallocated.
Let me see about going back to questions for both Robin and for Prabha. Prabha, you have been directly involved with fungal therapeutic development. Can you go over specifically around what you've seen as the differences here on the fungal side? Because you have a storied history of antibiotic development. What do you see as the major differences between the two, please?
So the major difference between fungal and bacteria is actually a very important question. A bacteria is not a mammalian cell, is not a eukaryote, whereas it does not have a nucleus. It doesn't have all of the components a mammalian cell has. Whereas an yeast cell, a fungal cell, is a eukaryote, has all of the components our cell has, and a few differences. So it is fewer targets than bacteria. So we are all ready to get drugs which are targeted, which are those magic bullets which will only hit the fungus. There are only a few magic bullets one can use. Having said that, there are a few targets, and those drugs have been identified, such as the azoles. We are all familiar with fluconazole, amphotericin B. Amphotericin B was a very old one. It used to be called amphoterrible because it's very toxic. But it's still used because that's what we have to resort to. Now, fungi are found in the environment. They grow on plants. They're happy in the environment at a lower temperature than our body temperature. But they do infect people as they get immunosuppressed. So if they're sitting on your skin and they can get inside you, they find healthy media for it to grow. Now, to do a trial in these patients and develop a drug, to develop non-inferiority to these older drugs, you have to show clinical efficacy, which is non-inferior to the existing drugs. So to do that, you have to do larger populations. And there are not that many patients who, thankfully, get these fungal infections. So it takes a long period of time. Could take 8 years to meet these new guidelines or longer. Some of them have taken 10 years, which are still running. And then on top of that, if one looks at all-cause mortality, some of these very sick patients may die, and that would be held against you in your trial. Then to get selectivity, so you cannot have many side effects because many of these targets are shared with human cells. You have to get selective compounds, and yes, we have some. We have some new ones, but majority of them are the same class as the old ones. And we need new compounds and new targets in fungi, something which is cidal because these patients don't have an immune system, which is very good. So you need fungicidal drugs, which will work very rapidly in these patients. Now, Candida auris, which is the topic of this conference, is actually very difficult because it is already resistant to the azoles. It started that way. And it might be because we've used a lot of azoles in the environment as in agriculture, but we don't know why, but they are resistant to azoles. They were very happy living on plants. They are unlike some of the other fungi, they like higher temperatures. And so they can actually survive, at our body temperature and at slightly higher temperatures. And so they're very happy sitting on our skin, but can get inside us. And they're resistant to the azoles. Luckily, some of the new compounds are working against them. But for how long? Resistance is already happening. And we need to find new targets and new molecules.
Got it. Thank you, Prabha. Dr. Thomas Walsh has just joined us, and I'd like to start by saying Dr. Walsh represents the front line in dealing with infectious pathogens, and specifically Candida auris. So with that, Dr. Walsh, can you please just tell us about yourself? And I want to dive in with questions specifically with you.
Yes, thank you, Steve. Are you able to hear me?
Yes, we can.
Very, very well. Great apologies. I was in 14 minutes before start, but we ran into some major IT problems. Nonetheless, first of all, I want to thank Steve and the other and the moderators and my colleagues for inviting me to participate in this important symposium. I think it's important to take a broader perspective and understand host and diagnosis and pathogen, and to which I've devoted really much of my life. And from the time that I was a student at Johns Hopkins and going forward, was struck by the devastating effect of invasive fungal diseases, losing many of my patients with severe underlying conditions, neoplastic processes, for example, to invasive fungal diseases. Having graduated from Johns Hopkins, spent 10 postdoctoral years, half in the laboratory, half at the bedside, boarded in infectious disease, oncology, and medicine, going on to National Cancer Institute for nearly a quarter century, and spearheading a major effort for studying invasive fungal diseases from bench to bedside, having contributed to the development of 14 antifungal agents, as well as several other antibacterial agents, both in pediatrics and adults, from the laboratory to the bedside. In the laboratory, we studied innate host defenses, in vitro and vivo pharmacology, as well as much of the metabolism and drug-drug interactions that we encountered, as well as rapid molecular diagnostics, a number of platforms of which we've innovated and developed. Fast forward then, I founded and developed the first transplant oncology infectious disease program at Weill Cornell Medicine, New York Presbyterian Hospital, and more recently was recruited now to build the Center for Innovative Therapeutics and Diagnostics, whose mission is basically to spearhead the next generation of antimicrobial agents and rapid molecular diagnostics in innate host defense augmentation. If we look at Candida auris, and well, the epidemiology, as we just heard, is profoundly compelling. What strikes one especially is the devastating impact that it has on individual patients. Just very briefly, to humanize the tragedy of this rapidly expanding pathogen, take, for example, a 55-year-old gentleman in otherwise good health, but had some cardiac dysrhythmias for which he had a pacemaker, came into the hospital for just a routine surgical procedure, developed Candida auris fungemia, with that landing on the pacemaker wires. The ultimate consequences, the whole pacemaker, everything had to be ripped out. He underwent several more complicated operative procedures, eventually survived, but only after considerable suffering and pain. Another situation, a 62-year-old gentleman out of state came in for procedures leading to necessitating nephrostomies. Those are percutaneous catheters that will go into the kidneys. He had long-standing kidney stones, nephrolithiasis, and this was thought to be the best way to relieve the obstruction. They became infected with Candida auris, and he underwent nearly a year and a half back and forth for treatment. The problem is the first-line agents that we have against these organisms, called the echinocandins, that would be, for example, micafungin, inidulafungin, caspofungin, and at that time we did not have razafungin, but that is also another echinocandin. They're extremely potent, highly fungicidal. As Dr. Fernandez says, we do need these fungicidal agents. However, this class of molecules, because they're so large and largely hepatically excreted, only have a small release into the kidney. And so consequently, it was more than a year and a half in which we could finally eradicate the organism after multiple hospitalizations, disruptive of his family life, disruptive of his work life, not to mention the cost as well as the pain, the suffering that this poor man underwent. Eventually, we eradicated them. But one is only struck by the limited opportunity to be able to manage these. And finally, underscoring the critical nature of diagnosis, a gentleman was hospitalized with complicated intra-abdominal surgery, colorectal surgery for cancer, but which would have gone relatively well had it not been for the candidemia. Unfortunately, the candidemia 1 was not recognized early through no fault of the laboratory, just because of the paucity of our rapid technology, relying upon culture-based, where we know that organisms can penetrate through tissues, be in patients throughout extended periods of time before it finally comes into the bloodstream, and then the culture-based technology is even then delayed when it does become positive. And then with that, the organism was not recognized as an Auris initially. This organism can mimic in the laboratory on the plate a number of other organisms, such as Candida glabrata. It's not a germinating organism. Finally, it was identified, but not until extended lengths of time had transpired, resulting in delay in diagnosis and a very complicated process. Ultimately, this poor gentleman developed multi-drug resistant, where the organism had become completely resistant. And to that note, if we look at the CDC's commentaries in the most recent Annals of Internal Medicine paper, they're describing a threefold increase now in echinocandin resistance. And in conferring with my other colleagues in other countries, for example, in India, they're all where it dominates in some hospitals as the primary pathogen amongst the different Candida species, even exceeding albicans and parapsilosis. They are now seeing multi-drug resistance such that effectively they're out of medicines.
Dr. Walsh, I want to just get a quick your opinion, and then I want to turn to Dr. Robinson. Can you tell us specifically about how difficult it is, as far as when you treat these patients, to test and then clear them when you've treated them? How burdensome is that? And if you could just answer that quickly, because I do want to get to Dr. Robinson with something that he mentioned earlier.
Yep. So clearly, in the broader context of the detection of Candida, this is an organism that even with the best of blood cultures from autopsy-based series, a large series in which we had published, where patients tragically had died from invasive candidiasis, more than half were not detected by the state-of-the-art blood culture detection system. The organism can extend widely into tissues before it spills into the bloodstream, and often when it's in the bloodstream, it's only the tip of the microbiological iceberg. There are molecular diagnostic systems, but they're vastly underutilized and not widely available in most institutions. So we have, for all organisms, all Candida species, a paucity of platforms, and we're challenged with the ability to rapidly diagnose this organism as this invading tissue, but has yet to be present in the bloodstream. Once we identify the organism, and delay in diagnosis is clearly associated with higher mortality, once we identify the organism, then the question is how long to treat. Now, most of the candidemia clinical trials have been originally designed for a 2-week course of therapy. That probably will work well for most cases of uncomplicated candidemia. But for deep-seated candidiasis, complicated candidiasis ranging from intra-abdominal, cardiac, as I described in my first patient, central nervous system, there's vast uncertainty as to how long to treat. And we often underestimate in complicated surgical patients the extent of disease. So having a biomarker that could allow us to say you've eradicated the disease would also be a very powerful tool.
Got it. Thank you. Dr. Robinson, you had mentioned something in a couple of conversations as far as the linkage between what we've seen with COVID and what we're facing right now. Can you describe that and talk about how that linkage you see, what it means for us today?
Thank you, Steve. And thank you, Dr. Walsh. That was an excellent summary of what you're seeing in the front line, and I appreciate what you're doing. The COVID-19 actually has a huge number of ripple effects, whether they be in the economy, society, and at large, but also on how we manage other diseases. And since there was sensitive patients for COVID-19 infection or individuals with immunocompromised, you actually then have an overlap with those that are at real risk for the new epidemic with Candida auris and other species. And that said, with the next gen COVID-19 initiative that the Biden administration announced recently, this actually could be part of that HHS is launching this summer, in fact, and that it can dovetail with the announcement last December that BARDA finally after years of pushing towards the policymakers, being able to fund the development of new antifungal agents, put a program together, and hopefully this year, 2023, those awards will be made, and we can actually get several new classes of antifungals out there into development. That said, though, I worry that we are going to have a reprise of what Julie Gerberdine said in November of 2019 at a meeting in which she went back through her records and when she was CDC's director and brought it all the way to 2019, pre-COVID-19 Prussian comments, is that we go through the lessons learned, and she said, I've gone through all the lessons learned, and they're exactly the same every time. And this epidemic is not going to wait because the number of patients from COVID-19, long COVID-19, are going to be identified, and they're going to be around for a long time, and they're going to be susceptible to these hospital-associated infections. And the requirements to control this are not unlike what we saw in 2020, 2021, and 22 that had to be done to bring the COVID-19 pandemic. So there's some lessons learned there. There's a little bit of hope there, but I really hope that the lessons learned do not include Candida auris. And we should push the Biden administration to include heavy attention to this disease, similar to what the Obama administration did with antimicrobial drug resistance with the CARB initiative. That actually came, after many, many years of the CDC warning of antimicrobial drug resistance, is that, we were at the last line with vancomycin. It was about to go under. And we I think, in a similar situation now with the 3 classes of drugs for the Candida species. Thank you, Steve.
Well, thank you, Dr. Robinson. Prabha, Dr. Fernandez, it looks like you're going to have the last word here. Since you're overseas, does the world understand what we face and what we're looking at right now? Not just the United States, but the world. And you obviously tied it in by mentioning the mortality rates in India. So what do you think? And with that, you've got the last word on this panel.
And so, what's happening there will happen here. We saw that in COVID. So, it's going to come. It's just that you see what's going to happen. You just get a crystal ball to see is basically what's going on over there. In fact, people are afraid there to go into surgery because they're going to get into those infections. And even for essential surgery or even essential cancer treatment, there's reluctance now to treat them because they could get one of these infections. So if that happens here, that is, and it's coming. It's just a matter of time. And I think that's what's important. One point I'd like to make is that, we all think Candida auris is a new disease. It's not a new disease. We learned you find what you look for. Candida auris actually was identified a while ago, even in blood, and it was just stored away, was misidentified. And after first detection in 2007 in the year, suddenly you found that there were 3 clades in 3 different continents. So it was happening. We just missed it. And then we found it in America when we started to look for it in 2011. So we are coming to a perfect storm. You're having increasing resistance rates, increasing immunosuppressed patients worldwide, various diseases, and you have this bug which is now adapting to higher temperatures as climate change and higher temperatures, and they're adapting to it. And this bug is very sticky, as we had heard. It sticks to everything. It's very hard to get rid of. And the moment you have a lesion, it gets in. The time is coming, and we can't say it's happening there. It's going to happen in the United States.
Got it.
Steve,
I'm sorry.
One point. it's very interesting that at the beginning of the HBO series, the last days, that they recognized a panel that had talked about fungal infection worldwide being epidemic and pandemic. 25 years before that, shows apocalypse occurred. I hope that next year that we are not the panel that's actually being on HBO's Last of Us. So we have to move forward, because we were saying the same thing as it was in that very first episode. Thank you.
Robin, I've known you for 20 years plus. This is the first time you've made me laugh, but it's not a good laugh. So thank you. With that, Sarah, I turn back to you.
It's a laugh of concern, truly a laugh of concern. It's really, if you think about it, it's a bit terrifying, the landscape that you're introducing to all of us. So Steve, Prabha, Robin, and Tom, thank you for joining us for this section. We will move on to our next session. And those of you who can stay, please do come back as we move into Q&A. So we're going to bring on our next panel lineup. Are we ready to do that, everybody? Carita Anderson. Well, our next panel.
Hi, Sarah.
Oh, okay. Is everybody coming on? Okay, I'll introduce you all. Okay, so I'm pleased to introduce our next moderator for the second segment. That's Carita Anderson. She's editorial director and a biopharma expert, and she will be joined by David Angulo. He is Dr. David Angulo. He's president and CEO of Synexus. Dr. Michael Kurilla, he's the director of NCATS at NIH. And John Spurzl, president and CEO of T2 Biosystems. So thank you all for joining us in this little complicated dance, but I think your message is really important. With that said, Carita, I'm turning the virtual microphone over to you, and I'll go off screen for a while and come back in for Q&A.
Got it. Thanks, Sarah. So that was definitely a really interesting conversation, and Steve, Robin, Prabha, and Tom really covered a lot of ground. So David, Michael, and John, we have our work cut out for us. Before we dive in, I will do just a really quick round of introductions so that we can jump right into the topic. Like Sarah said, my name is Carita Anderson. I've spent almost 2 decades in the news business covering the biopharma and healthcare industries. And today I'm joined by John Spurzl, President, CEO, and board member at T2 Biosystems. Prior to T2, John was president and CEO of ChemBio Diagnostics. And before ChemBio, he served as president and CEO of ITC Medical. Previously, John held executive and commercial leadership positions at a number of companies like Access Shield, Bayer Diagnostics, Instrumentation Laboratory, and Boehringer Mannheim. Michael Kurilla also joins us today. He is the director of the Division of Clinical Innovation at NCATS. He oversees the Clinical and Translational Science Awards program. And before NCATS, Michael served as the director of the Office of Biodefense Research Resources and Translational Research within NIAID. And prior to NIAID, Michael was an associate director for infectious diseases at Wyeth. He has also worked at DuPont and at the University of Virginia Health Sciences Center. Lastly, I'd like to introduce David Angulo, who is the president, CEO, and board member at Synexus. David joined Synexus in 2015 as Chief Medical Officer, and previously he was Vice President, Research and Development of Brickell Biotech. Prior to Brickell, David held several senior positions at Stiefel Laboratories, including head of clinical and medical departments. David was also responsible for several anti-infective programs at Schering-Plough Research, and brings his perspective as a clinician since he was an infectious disease physician in a pediatric hospital. Okay, so let's get started. The first segment gave us a really good overview of how we have got into the state that we are today, and in particular, what we are seeing with C. auris and this latest outbreak that we are facing. So in the second segment, what I hope to do is pull on some of those threads and identify the gaps and try to come up with a holistic approach to how we deal with this problem. Now, before we talk about all those solutions, I do want to talk a little bit about the risks that we face again. The first panel certainly laid that out clearly for us. But just to talk about the risks that we face from emerging pathogens, and importantly, the risk that we face if we do not tackle this situation. So, David, I might start with you. And to begin, this is a bit of a biology 101 question, but I think it's an important one to help us all wrap our minds around just how big of a problem this is and just how real of a problem this is. And what I wanted to ask you is if you could describe how new pathogens emerge and how those reasons for new pathogen development aren't going anywhere, in fact, are probably increasing, and how all of that informs your decision-making when it comes to therapeutic research.
Thank you, Candida. Thank you to the organizers for the invitation to the panel. And certainly this is a fascinating question. Certainly we start learning about the new pathogens that really, take an example of Candida Auris, that we started learning about that particular pathogen about 2 decades ago, so to speak. And then at the beginning, the numbers were small. However, we are a global organization that now we are hearing and we're seeing that the numbers are increasing and they are taking the role, or they are taking the place than other pathogens we used to see. In the past, we have been seeing invasive candidiasis for many, many years, decades. Yeah. The vulnerable population is increasing. So you have greater number of people that are exposed and potentially vulnerable to these pathogens. And now in the past, Candida albicans was the dominant pathogen. We started seeing years ago that Candida glabrata, another species of Candida that is also often multi-drug resistant, started taking over a number of cases and started growing. Now we see Candida auris. At the beginning, it was few cases here and there. And now we are seeing that many institutions, as we heard, Candida auris is becoming the predominant pathogen because it's much more difficult to kill. It's way easier to spread. And so the opportunity for that to be the dominant Candida species within a particular hospital setting is very, very concerning because we have very, very few tools to really treat fungal diseases. That information is critical for us as researchers and drug development. And the challenge is that we need to develop drugs. A drug development process takes 7 to 10 years. We need to develop a drug that is going to be useful 7 to 10 years from now. So we really need to be understanding what are the trends, where is this going. As an example, the drug that we are developing right now, we started doing clinical research in Candida auris 4 years ago because we knew that even if the cases were very, very small there, it had all the characteristics to become a real global program. So yes, this information is really what will drive us to really decide next steps in development for new therapeutics.
Yeah, David, thanks for laying out that evolution of the Candida species because, it definitely feels like 2000 has been marked by emerging pathogens, right? Indeed, HIV appeared last century, but then fast forward to this century and we had SARS in 2003, swine flu in 2009, MERS in 2012, Ebola in 2013, Zika in 2015, and then, of course, COVID-19. So, Michael, how are the pathogens overall? How are they changing? What do we learn from that? What do we learn from the pace of past outbreaks? Does one feed the next? Robin from the last segment spoke about how COVID-19 and C. auris are linked. I wonder what your thoughts are on that, and to put that all into context of how real and just how imminent a future outbreak is.
Well, I think we first need to recognize that part of the reason for emerging pathogens is partly it's a consequence of our technology to be able to identify them. You can look back over the 20th century, and there's lots of examples of ill-defined outbreaks that were never fully explored in cases where the CDC actually has stored samples. They've gone back and we've identified what those cases were. It's partly a consequence of our technology to be able to do this so well. It's also a consequence of the increasing global population, both from a population density standpoint, so infectious diseases can much more easy to pass them around, coupled with the fact that we have a relatively interconnected global system, which means that something that occurs as a local isolated outbreak in one country can very quickly, in a short amount of time, be transmitted throughout the globe. If you go back to the 1889 pandemic, which was a major global pandemic, still some arguments about exactly what the cause of that was, although historically it had been regarded as an influenza pandemic, but in retrospect, there's been quite a bit of recent evidence that suggests it may have actually been another entry of another coronavirus into the human population. But in 1889, that was pre-aircraft, pre-automobile. It was basically steamships and trains, but that was enough to allow it to encircle the globe in about a 4-month time span. So I think it's partly a consequence of our interconnected global situation. And the other, I think, is that we have a very pronounced relatively advanced, at least in throughout the developed world, in terms of medical technology. So as David highlighted, we have a much larger population of vulnerable people who are immunocompromised for various situations, transplant situations, cancer therapies, high-dose steroids. Diabetes is also a very rampant medical issue that predisposes to a lot of immune issues down the road. So we have created a much more at-risk population based on our ability to treat diseases that we never would have really been able to address adequately in the past. Now, the other issue I think we are also experiencing, and Prabha touched a little bit upon this, the question of the origin of Candida auris, where did it really come from? Yes, there was some failure to recognize it before it was officially recognized as an outbreak. But the fact that there seems to be at least 4, perhaps 5 different distinct clones that have emerged in different geographic regions does suggest this is a much more environmental kind of phenomena. And one interesting hypothesis that have been raised is that since many of these fungal pathogens typically exist in nature, they're not as well adapted to human or mammalian body temperatures of 37 degrees centigrade. But with global warming, they are actually adapting to global warming and are becoming much easier to actually colonize and infect and invade human as well as probably other mammalian species. And so, we've seen in the past, I've been at NIH long enough that, we typically get asked a question from Congress, what are the health effects of global warming? And, one of the things we always spoke about was changes in distribution patterns of mosquito populations that would change where certain types of diseases we already knew would be prevalent, that they would be moving further and further north. But we may be seeing in Candida auris the possibility that global warming is actually changing the way fungal pathogenicity actually operates, and that's quite concerning, I think, and really highlights the need for more intensive therapeutic development. The last comment I'll make is that this Candida auris is not unique in this regard. There was a recent study that was done by the Washington University in St. Louis that was basically looking at largely Medicare claims and came to the startling conclusion that historically, at least what I learned in medical school, which granted was a long time ago, valley fever being from the southwest section of the United States, it's in fact far more diffuse throughout the United States. It just hasn't really been recognized. And so I think, again, echoing back to Prabha's comments, is that we just simply haven't recognized the full extent and distribution of fungal diseases. And as a result, they have not gotten the attention that they really deserve.
So, John, you're a diagnostic expert on this panel. So in terms of what Michael started off his response with and ended with, the importance of recognizing these pathogens. And I mean, also, I would like for you to, comment on that factor, but I also wanted you to comment on the fact that, yes, past outbreaks have largely been due to viruses, but clearly, we have reason for plenty of concern with fungal infections, and antimicrobial resistance has been an issue for a very long time. So when we're thinking about outbreaks across pathogens, what do we need to keep in mind when it comes to developing diagnostics? How does that inform diagnostic development?
Well, thanks, Karita. I speak as somebody who's been involved in the development and commercialization of rapid diagnostics for 3 decades. And a couple of the outbreaks that were described by Mike was Ebola and Zika virus. I mean, many of these pathogens present with similar signs and symptoms. Ebola looked a lot like malaria. Zika virus looked a lot like dengue fever and chikungunya virus. And so detecting and identifying the specific pathogen and its potential resistance characteristics is critical to achieving targeted treatment. And in my view, it's critical to unlocking the power of targeted antibiotics and antifungals like those developed by David's firm. So for patients that are at risk of sepsis due to a bacterial or a fungal pathogen, including Candida auris, rapid diagnostics are critical. Data shows that for each hour of delayed targeted antimicrobial treatment, the risk of death can increase by up to 8%. And so from a product development perspective, we need to focus on rapid diagnostics that do not rely on a positive blood culture as the clinical specimen, because blood culture by itself is notoriously insensitive. So it will miss a lot of infections, and it takes a long time, anywhere from 1 to 5 days, 2 to 3 on average.
Yeah. And Prabha was certainly making a very similar point too. So, what I would say right now is we know what the solutions are, right? We've known what the solutions are for a while. We knew what the solutions were pre-COVID-19, but yet we weren't really fully able to implement even what we did know how to deal with a pandemic. Forget about the preparedness that should come before a pandemic. What needs to change in our thinking so that we start to not just, we move from trying to draw attention to this problem to having government and industry actually focus on this problem? And John, I'll come back around to you for this one as well. Why the stalemate in funding? Why the stalemate in implementation? Sonny, talk about it specifically. You just started to, during COVID-19, one of the things that definitely didn't work as well as it should have was the role of testing and diagnostics. Why didn't that happen? And how do we make sure that that does happen in the future, not just during an outbreak or a pandemic, but also in our day-to-day medical system to, identify and diagnose and track these pathogens?
This is probably going to sound harsh, but I believe it's a lack of leadership. And I'll share a specific example. Our company has the only FDA-cleared diagnostic tests that are able to detect bacterial and fungal pathogens directly from whole blood in just 3 to 5 hours without the need to wait days for a positive blood culture. In fact, one of our diagnostic tests is a T2 Candida panel that identifies Candida pathogens. It's FDA cleared, and it detects approximately 90% of Candida species. And we plan to add the detection of Candida auris. We already developed a Candida auris swab test with funding and support from CDC, and we've converted that to a direct from blood test. And I think here's where the lack of leadership comes into play. I personally contacted BARDA, with whom we have an active product development contract, to request funding to complete Candida auris development. and a US clinical trial. And I was told, quote, fungal pathogens are not in BARDA's mandate, end quote. I mean, as taxpayers, healthcare providers, and consumers, that's not an acceptable answer. We have to do better than that. We have to demand better than that.
Yeah. David, I'll pose a similar question to you from the context of therapeutics. You know, one of the biggest challenges has obviously been a lack of business interest. And we heard this as well in the first segment about, you know, the thinking behind the ROI when it comes to antimicrobial investment. How do we change that situation, or other challenges that you think are critical for us to talk about and try to figure out how to move past to ensure that we have more focus on antimicrobial drug development?
Sure. Thank you, Candida. I think that let me just start by saying that certainly for Candida auris, there are three, to be able to control this particular disease, there are three key components, in my opinion, that we need to invest and we need to focus in the three of them. One component is important, and we're not talking about here in this panel, but it is hospital control practices. So adequate hospital control practices can somehow limit or minimize the impact of these new pathogens have in spreading within the hospital. That should not be ignored, should always be part of the equation. The other is adequate and accessible diagnostic tools so that you really know what you're treating and that you really know the extension of the disease and the spread of the disease and which populations are being affected by. And the other area is adequate therapeutics that really have the adequate options. In this particular case, I don't know if it is known by everyone listening to us, but antifungal therapeutic options are very, very limited because of all the aspects that Prabha was mentioning. So developing antifungal drugs is much more challenging than developing antibacterial drugs because you really need to find a target that is adequate to kill the fungi, but it will not have toxicity against human cells. So that is a way challenging approach. And that is the reason why there are only three main classes of systemic antifungal agents that can be used to treat these very fatal infections. And I've been privileged to work in many of those classes because my whole career has been focused in antifungal development. And I would say that I've been privileged and fortunate to really being able to bring the fourth class. The first product of the fourth class right now already approved in the United States, not for invasive fungal disease, but for vulvovaginal candidiasis, what we are studying for invasive fungal disease, invasive candidiasis as well. But the challenge is exactly as you mentioned. We are a public organization. We really need to reach our investors in order for them to believe in the future that this will represent a return of investment for them. Because otherwise they will not put their money into supporting the development of new therapeutic options like us. So we have been, I would say, fortunate to be able to have, I would say, believers that have supported us through the development of this particular product. And we recently were able to create a fantastic partnership with a large pharma company that is now going to be commercializing that product. So in some way, it was a successful story. We developed it. We are continue developing, and we are finding right now someone that is believing the story and being able to invest in the commercialization. But this is not the norm. I would say that this is not what is happening every day, and it's not after a few years of a struggle, many years of a struggle. So in order for us, we really need to think about solutions right now. Certainly, having some push and pull incentives are extraordinarily important, but also from the regulatory point of view, from the approval of drugs point of view, we need to be way more open to consider less than traditional development paths for these kind of conditions in which it's very difficult to find sufficient number of cases to demonstrate really activity in large numbers. We need to use the tools that we have very well developed over the past years, translational medicine. We need to rely more on that. We need to rely more on the activity of the antifungal agent in other similar conditions. And then to really make a better understanding of what is the weight of evidence that in order to get approval of a drug, not necessarily via the traditional design of two well-controlled clinical trials all the time showing non-inferiority or superiority. That is a path for acceleration of approval of a product that we can all continue monitoring once the product is out there, if there are additional elements that we need to generate additional data on. However, the approach needs to come from multiple areas. Regulatory bodies facilitating development and approval, push incentives, pull incentives in order to facilitate companies that are not necessarily well-funded to be able to move to the next stage. And certainly investors will start believing more and more in those stories and will start putting a little bit more money and support into these development activities. We need to, all these pieces needs to be in place in order to really facilitate. It's not lack of scientific interest, by the way.
Right.
Not lack of scientific interest. We just had the Candida and Candidiasis meeting, which is an entirely group of scientists focused in Candida and biology of Candida and really trying to identify new targets for new antifungal agents. Who's going to pick up on those scientific discoveries to try to translate them into a deliverable therapeutic? It's a challenging story. So it's really going after those and being able to put all the money behind and intellectual design behind to try to make them into real therapeutics. It's a challenging part to really make that decision. But it is not lack of scientific interest and scientific and technology to identify new targets.
Yeah, and we've seen the FDA be creative in what they allow in terms of clinical trial design and outcomes for other diseases, even during the pandemic for the vaccine. So there is certainly a conversation to be had, and hopefully we have that conversation before we're in a pandemic situation and think about those creative strategies. Michael, David hinted at that third leg of this discussion and that third piece of the puzzle being transmission control. And I was wondering if you could bring us to that part of the equation just a little bit, from data collection to understand spread patterns, to better practices in healthcare systems. How do we see this important part of the solution through? And in fact, diagnostics play a role in this as well, in transmission control. Vaccination also has a role to play in transmission control. Is there a danger of over-reliance on a vaccine strategy? Is there anything as too much reliance on diagnostics?
Well, I think, echoing what John and David have said, I think one additional component that makes Candida auris very concerning from my perspective is that not only is it a severe disease to those patients who actually contract it and have an invasive, but the ease at which the organism can colonize people's skin means that it becomes very easy to transmit throughout, such as a healthcare facility or any large congregate living situation. And this really highlights that, one of the issues we have to deal with here is a sort of disconnect between our healthcare and public health. They're really not connected very well. And so while we think about diagnostics for making a clinical decision on an individual patient, that's how you get reimbursement, unfortunately. But the same diagnostic technology or similar aspects of the diagnostic technology can be applied to public health surveillance, and there's just no mechanism for really doing that unless someone like the CDC or a state public health system actually takes it on. And so we're really, typically, when you have these sorts of situations where something new develops, we are really although we're not lacking in the technology to develop the tools, we're lacking in the mechanisms and the funding to actually take full advantage of that technology to actually provide not only the individual patient care that is necessary for those who contract the disease, but the public health surveillance aspect to really stay on top of it and limit it. Now, vaccines would be a very ideal solution as a part of the armamentarium. Fungal vaccines have been, there have been efforts for a long time. It's still a scientific nut that has to be cracked at this point, I think, before we can really fully move in that direction. So the therapeutics, the rapid diagnostics and therapeutics, I think, is where we are right now. So.
Yeah, definitely. Well, thank you, Michael, David, and John. That was a lot to go through. And of course, I have, 500 more questions, but so does the audience. So I see Sarah back on, and I'm sure she'd like to walk us through some questions from the audience as well.
I think. Go ahead, Mike.
Sarah, before we talk about the questions from the audience, we actually took a polling from our online polling, and the priority was a discussion about the diagnostics, specifically what needs to be done next on diagnostics, because we're seeing treating without understanding. So I think, John, that question falls directly into your sweet spot. Can you answer that?
Okay, well, you cut short my thanking everybody for the second panel. So everybody for the second panel, consider yourself thanked. Okay, Steve.
Well, thank you. I think that there are things that we need to do, specific actions that we need to take to facilitate both the development and clinical adoption of diagnostics. And those things include allowing the use of contrived samples during clinical trials, especially for these emerging pathogens. It's otherwise very, very difficult to find them in a standard clinical trial. It would include changes to guidelines and reimbursement. Those are key elements to facilitating clinical adoption. The good news is these are all things that were done during the COVID-19 pandemic. So there's nothing new here. We have the playbook. What we have to do is we have to take proactive actions. And as I said before, from a diagnostic perspective, those actions need to include enhancing the current care protocols through the use of culture-independent rapid diagnostics.
Sarah.
Okay.
Sorry.
Okay. Is the show mine? Is the stage mine again?
It is back to you.
Okay, so we have some questions that have come in from our audience. And so let's start with one of the questions is that drug resistance was mentioned. Do fungal pathogens mutate more or less quickly than bacterial or viral pathogens?
So you want to take that? Oh, go ahead, Tom.
Are you able to hear me?
Yes, we can, Tom.
Okay, very good. The Candida species replicated approximately once every 2 hours. Bacteria will replicate at approximately E. coli Staph aureus at once every 20 minutes. The propensity for bacteria to rapidly become resistant, you can demonstrate in vitro with just serial dilution within 24 hours. In patients, we can see the rapid emergence of resistance, often in either a heteroresistant population or in the setting, for example, of Acinetobacter, which may already have an integron, where under pressure it may come in as a community-acquired pan-susceptible organism, but just under pressure rapidly within 24 hours expressing a wide array of resistance genes. Enterobacter, for example, having a stably de-repressed beta-lactamase. Other organisms can transfer plasmids very rapidly to become resistant to beta-lactamase, fluoroquinolones, aminoglycosides. Fungi take longer. Many of the organisms over time can be selected in a patient, but over the course of time, you may not see that emergence of resistance until 1 to 2 weeks, the example being Candida glabrata emerging, for example, to fluconazole. It takes much longer. However, the other challenge is that we have an emerging population of organisms that are now intrinsically resistant. So here you have Candida auris intrinsically resistant to the azoles. We haven't talked about them, but there are molds now that are intrinsically resistant to the azoles. So this is a major threat, and this is where the environment, where you're finding a lot of these organisms interfacing now with our immunocompromised population, are seeing much of the intrinsic resistance, and adding on to that burden over the course of time, a complicated ICU patient in the span of 1 to 2 months receiving antifungal agents. Those organisms will have enough time in which to develop resistance.
Thank you, Tom. Does anybody else want to comment on that?
Sure, this is David. So the other thing that is a little bit more complicated in this situation is that the treatment duration for most of the fungal diseases is longer than the typical treatment duration for many bacterial diseases. So you're talking about here several weeks, sometimes months, when there are deep-seated infections. And also it's more difficult or more not that common that really this kind of fungal pathogens develop resistance very rapidly. They do have greater opportunity to do so while on treatment because you are exposing them for many months or for many weeks to the antifungal therapies. So while the patient is on therapy, it is possible that they develop resistance to particularly to the azoles, etc., while they are on therapy as well to the echinocandids. So also it is less frequent. It is also concerning. But as I think that as Tom was mentioning, probably more concerning is those pathogens that have already an inherent resistance to a particular group of antifungals, because as we mentioned before, you just don't have too many options. So once you develop, and I don't know if we mentioned this, that among these three primary groups that are used to treat these very deadly fungal diseases, only one has an oral option, which means that if you're going to be receiving therapy for 6 weeks, and your bag is resistant to azoles, you are giving treatment for 6 weeks with an intravenous formulation. So those are very limiting treatment opportunities and treatment options for the physician and for the patients to deal with. So it's a little bit more complicated from that point of view. Treatment duration is longer, options are fewer. So development of resistance against one class is a big deal. It's a big problem. It impacts substantially the opportunities for treatment.
And I think, David, building upon your comments on the need for intravenous therapy, this is not just an innocuous situation. You have then by necessity the implantation of a long-term catheter, and you've now opened up your patient to the risk for bacterial infections ranging from staph to gram-negatives, further complicating their care.
Yeah, and let me just add, there's another logistical question here, and that is, while you could do infusion intravenous as an outpatient, we actually have very limited capacity for outpatient infusion capacity. That was something we experienced with COVID. The monoclonals that were authorized by the FDA, there wasn't a lot of availability, and the most outpatient infusion capacity we have is in oncology, and they actually don't want to see patients with infections coming into their facilities. Yes. So it is actually a real logistical a nightmare if you have to think about treating infected patients by IV in an outpatient setting.
Wow, it's
just a point, if I may say, you're talking about adults, which you can at least put in these catheters, infuse for long periods of time. And you think of babies and neonates, 2 hours is too long, and the volume of fluid you can give is very little. So there's an additional problem when these young babies are now getting sick with Candida auris.
So we have a few more questions to go through, and then I'd like to come back and ask, maybe a general or closing question of everybody. One of our other questioners has said, if we detect the colonization of Candida auris, clinicians can target C. auris for a likely cause of infection. Where are we in the development of C. auris colonization tests with FDA approval? That's an open question to whoever knows the answer to that.
Well, a lot of people are colonized with Candida auris. Does not mean it's going to cause an infection. I might have one, and I'm sitting here in Geneva, right? So, detecting it is not cost effective at this point. So if you get a patient who's going to get operated, then you could perhaps test them for Candida auris. But it still doesn't mean they're going to cause an infection.
So it's really not a practical question.
It's not a practical, not a cost-effective thing to do. It's there very often. It's very often on skin. We've got numbers. The numbers of C. auris bloodstream infection increased threefold, from about 500 to 1,500. But the number of colonization is 92% in the hospital. So what are we going to do? You might as well guess that they're positive.
Wow.
Sarah, if I may, the colonization question is one that's very, very regional and often very situated to individual hospitals. there are many hospitals in the United States that have just simply not seen Candida auris. Many other hospitals are awash with the organism, and some fall intermediate. For those that have a heavy burden, surveillance becomes operational because they need to know how to contain it. The organism is highly transmissible, and there are major infection control, hospital epidemiological policies, standard operating procedures for identification and isolation. Decolonization then becomes another concern, especially in compromised patients. But the resources needed for rapid and efficient surveillance are very cumbersome. So you're taking multiple cultures. You have to have technologists and examine those plates. The need for a rapid molecular screening technology is really paramount when you have those types of situations where you need to survey to identify patients in order to implement effective infection control.
Well, the problem is
Mike?
so daunting. Mike, you were going to say something?
Yeah, the other thing I would add is that I think from a regulatory standpoint, it is a major hurdle, probably an order of magnitude beyond the typical drug development, to actually demonstrate the clinical benefit of decolonization procedures. That's a very, very hard road to go down. And so I think a lot of companies rightfully just don't think the juice is worth the squeeze there.
Yeah.
Although Mike, I would add that there are a number of protocols, very simply chlorhexidine, that has been used. So off-label, there are attempts of decolonization, but unfortunately, they relapse. You try to decolonize, within 2 to 4 weeks, they've recurred again.
Tom, you mentioned something earlier on about how, the detection. I wrote a piece in Forbes 3.5 years ago talking about the detection that CDC had done for Candida auris. And it looked like a situation where only the hospitals who were sophisticated enough to pick up Candida auris, and some of them technically couldn't even culture back then the pathogens. So if you're talking about the leap from they can't even culture it to being able to check it in the hospital, it's just too great. As a matter of fact, the CDC map that we looked at the time literally were only the major teaching centers that had been able to come back. So if you're looking at the absence of the ability to detect it in people who literally have it in the bloodstream because culture takes so long, the leap to being able to clean out the hospitals is gargantuan. With that, Sarah, the closing question that you'd like to ask, because I don't want to.
I know we're rolling up on.
Before you change that, it's not that they cannot do it; they don't want to do it. Let's get that correct, because everyone knows how to do a culture. They don't want to do it.
So you're talking about structural barriers or a mindset, and mindset. Mike, I have a follow-up question. I think you'll be the point person on this, but as somebody who had an unidentified infection that had me in the hospital with IVs coming out of every arm. I know I'm not the only one here who's gone through that. The difficulty, they never identified what pathogen I had that was eating my skin away after I had had surgery. It's really terrifying out there. I know other people that have had to have body parts removed because they got some unknown infection. And I think, there's a lack of financial incentive for companies to do this. What about the government? And that's why I'm saying, Mike, you're probably the person on this. Is this not something, when we consider, the speed of travel, the rapid spread of pathogens from one country to another, native populations getting infected, where is the government in terms of both surveillance and supporting the research and companies that are focused on both the diagnostics and the treatment?
Well, I think, there's two aspects here. I think, partly we have a system where the way I would sum it up is, we'll pay anything for treatment, but the cure has to be free. and most of our anti-infective treatments are in fact curative in that regard. So they're not as remunerative as other things treating chronic diseases for the rest of your life. The other aspect, I think, is unfortunately the other sort of analogy we have with regard to healthcare versus public health is it's a kind of the leaky roof syndrome. You know, I can't fix the leaky roof while it's raining. But when it stops raining, the roof isn't leaking, so I'm off doing something else, and that's really what happens with public health in many ways. You know, I think that you know, the NIH in particular is very much focused on really trying to advance the scientific opportunities, but the expectation that for commercial products that would take advantage of that is really largely the expectations are that it's in the realm of the private sector to really move that forward. You know, and so the whole biodefense effort was really around the fact that there are some things that are just never going to have commercial interest. You know, whether or not fungal pathogens reach to that same level, I think they should, but that's well above my pay grade to make those decisions, unfortunately.
Yeah, and does anybody else want to comment on that? I think John, maybe.
I would love to weigh in on that, Sarah. As we're talking about fungal disease, CDC estimates the costs associated with U.S. fungal disease are as high as $48 billion annually. And CDC's called on health professionals to help lower the burden of fungal disease by continuing to raise awareness about both the life-threatening benefits of early detection and proper treatment. And that's what we've been talking about a lot today. I would argue that we're already facing a crisis with sepsis caused by bacterial and fungal pathogens. CDC also estimates that about 350,000 Americans die each and every year with sepsis. That's over 950 deaths a day.
And it's underreported. You don't know it, right?
Well, and that's 50 people in the 75 minutes we've been on this panel, just to put it in perspective.
Right.
And I think it's fantastic that CDC has identified certain pathogens as threats to public health, like Candida auris, and like bioterrorism pathogens that have mortality rates. You know, Candida auris as high as 60%, some of these bioterrorism pathogens as high as 90%. It's a great start, but we need to do more than raise awareness. We need to invest in rapid diagnostics, culture independent, that can help direct targeted treatment. That's the best way we address that. And that's the best use of the money to save lives, prevent bad outcomes, prevent amputations that you mentioned. That's all possible. We just have to demand better, and we have to put funding behind it.
Right. It's whose money? You know, the question is, where is the money coming from, right?
I think the money's there. It has to be allocated properly. I mean, 350,000 deaths. If CDC reported that in the top 10 causes of death, it would be number 3 behind cancer and cardiovascular disease.
Right.
COVID is transitory. I mean, we have to be honest about the issue that we're facing right now and put funding and support behind it, just like we did for COVID, and we can solve it.
John, I agree with you 100%. So before we sign off, before I thank everybody, Karita and Steve, do either one of you have final comments that you'd like to make?
I will just say that I think, you know, we've obviously spent time talking about the science and therapeutics and testing and transmission control, but and this was raised as well, the point of leadership and communication is really important here too. We saw firsthand during COVID how a lack of clear leadership and a proper communication really damages public morale and trust in what we're trying to do. And that's an effort that also needs to be thought through.
Thank you, Karita. Steve?
First off, thank you again, Sarah and Biotech Showcase for arranging this. I would say that we're in a unique situation here. We do have the answers to deal with these critical issues. We do have the ability to actually change things right now. It's not esoteric, it's not academic. The difference that we need to see is that people actually demand more than just go out there and say, well, wouldn't it be nice? That clinicians say, I'm not putting up with the status quo, because at the end of the day, ultimately the outcomes are not good. And I think that Now is an opportunity for people to go out there and say, this is what we can do right now, and taking that first step. And again, thank you, Sarah, and thank you to Karita and all of the panelists as well.
Yeah, Karita, Mike, David, John, Tom, and Prabha, and of course, Steve. Steve, we'd like to thank you and WBB Securities for sponsoring this. And I think it's the research group of WBB that is our sponsor. I'd like to thank our audience for participating and joining us. As I noted earlier, this salon will be downloaded and available for on-demand viewing a bit later on today. It takes us a few hours to do it. Before you all leave, we have a brief survey. We'd like to invite all of you to take part of that in that survey. And please join us at BioFuture coming up in New York City in October of this year. With that said, thank you all. We're signing off.
Thank you.
Thanks, everyone.
Thank you.
Bye.